Hi, Aparna. How are you? Good, good. Thanks. Yes. Yes. Thank you. Thank you very much.
Now it is. Yeah. Hello, Aparna. Can you hear me now? Hello, Luis. Yeah.
Hi, Dr. Charan. Thanks for the
How are you?
opportunity to share with you. Good,
Yeah.
good. And you?
I'm doing good. It's today evening at 7 p.m. for us in India. Weekend. People are joining slowly. I hope some more will join. Already several have joined. Hopefully, some more will join as we proceed. Yeah. People keep joining. Yeah. Maybe 2 minutes more. We are already 15, I think. 15, 20. Generally, we used to have attendance of 100 minimum. But this time,
Okay.
yeah. Generally, Louis, we used to have this at 11 a.m. in the morning. This
Okay.
time to suit your convenience because timing is very hard for you. 11 o'clock for you will be what time? What is the time difference? 10, 11 hours, 12 hours? 12 hours, okay.
Mhm. Yeah.
Mm.
Yeah, also in Costa Rica, for most people, on Saturday they don't work. So
Okay.
then also the attendance gets a bit complicated on Saturday.
How
Well,
is the weather?
I live in San Jose, and San Jose, the altitude here is around 1,200 meters. So
Hello,
this
yes.
means that the temperature, I would say it is around 22, 23. So it is
I'm
very
in
convenient.
Copenhagen now. Like, kind of,
Yes. Yes. How is
okay.
currently in your place? Is it very warm? Okay.
Mm.
Mhm. Okay. Yes.
Yeah, I think let us start, Aparna. Let us start. Yeah. Good evening, everyone, and good evening, Luis. Thanks, Aparna, for those few pleasant, nice introductory words you said about me. Now, as Aparna asked me, I have to moderate this session and introduce today's speaker, Luis Barboza. Dr. Luis Barboza-Barquero, I think I got the pronunciation right. He is an accomplished agricultural scientist and currently serves as the director of the Center for Grain and Seed Research in Sigras at the University of Costa Rica. He has received advanced international academic training from the Max Planck Institute for Plant Breeding Research, Germany, Wageningen University and Research, the Netherlands, and the University of Costa Rica. His expertise spans seed systems, grain quality, molecular plant breeding, and agricultural innovation, with a strong focus on advancing sustainable agricultural development through research, technology transfer, and capacity building. We're very pleased to have you with us, Luis, and we are all eagerly waiting to listen to your thoughts on the topic of today's talk. Please, the floor is yours. Yeah.
Thank you. Thank you, Dr. Aparna and Dr. Charan, for the invitation. And for me, it's a great honor to share with you, in your case, in the evening. So I also thank you very much for your time. And I hope it will be a nice way to start the weekend and also the rest time. Also, I would like to congratulate you for this initiative. Thanks for ATPBR. I think with all the introduction Aparna did, and also I was checking a bit the webpage and everything, it's like very good job that you are doing, and to open these spaces, especially for farmers, for students, for the productive system. I think it is very relevant to advance on agriculture. I will start sharing. If at some point, especially Aparna, if you can tell me if something is going on with the sound or something, just introduce me and then we can try to solve.
We can see your screen. Yeah, we can see it. Maybe you can make it full.
Okay.
Yeah, clear. Yeah, perfect.
Yeah, okay. So there is fine. Okay.
Yeah.
Good. So today, the topic, I would say it is very relevant, talking about climate resilient seed systems, innovation, seed science, quality, and global agriculture. It is something that we are really looking always for trying to improve our systems. I would like to set you a bit on what we do eat here in Costa Rica. On your left, you can see what is our traditional breakfast, and it is a breakfast that we use very regularly, and it's called gallo pinto. Basically, you will see that it's a mix of rice and beans, and also people, mostly they will add, for instance, a source of protein like eggs. That will be the most common. Sometimes they add plantains. Sometimes they will use sour cream. So there are some add-ons, but on the left, you will see that is the basic dish. And on the right, you will see what we call casado. That casado, it is a source of protein. In this case, you will see that it is like chicken. Then there can be egg, plantain, rice, beans. There will be a salad, and there will be chopped vegetables. And regularly, in most cases, people will repeat the same for dinner. So if you see, for us, especially rice and beans, it is something that we consume a lot. The per capita consumptions of rice, actually, it is 45, 50 kilograms per year. That is our consumption. So that is one of our key products when we talk about nutrition in Costa Rica. So this is a part of the map of the world, and here you can see where it is located, Costa Rica, and you will see that Costa Rica. Then on the north we have Nicaragua in the border, and then on the southeast then it is Panama. Then we have the Pacific Ocean that is on one side of the country, and on the other country it is the Caribbean side. Here you can see San Jose, that San Jose it is where currently I'm located. And you will see that all this part in the middle, it is a mountain that it is crossing, and it goes even through Panama. And the highest point, it is 3,800 meters above sea level. This means also we have very different weather conditions. The Caribbean tends to be very rainy and humid, and then the Pacific, then it is some part it is rainy, like the central part. The northern part, it is very dry. So then we have very different climates across all the country. Most people will live on the middle of the country. That is where the highest population is established. And the main production regions then will be located on the northern part, then also on the sides of the Caribbean part. That is where we produce most of our products, although we do agriculture in all the country. Most of the exports in Costa Rica are, in the case of agricultural products, are banana, pineapple, coffee. There are some companies that are seed companies, so they don't export that much volume, but a product that it will have an added value. When we go to food insecurity, here I can show you again the map of the country, and then you will see some percentages. This I got from a report from the Ministry of Health, and that is a report from 2021. But I spoke with experts from this topic, and they told me that is the best study that so far we have. So this mainly shows that there are some levels of food insecurity or prevalence on our country. Especially, you will see those located on the rural areas of the country. So, despite that we are a small country, we have also problems with people dealing with food insecurity. Also, in the case of agriculture, we have different challenges. In our country, in Costa Rica, we don't have, for instance, subsidies. We don't have strong supports for the farmers. There are some issues also dealing with insurances for some products. When it goes to loans in banks, sometimes the interests are not maybe the best for the agriculture. And also it has happened to us that there has been some decisions from the government that they have affected the production. The last case, it was rice, because rice we used to produce 40% of the rice that we consumed. But in the previous government, like 3-4 years ago, it was a decision to take out a tariff, and then when this tariff was taken out, then for the people it was like cheaper to import the rice. And then in this case, from being producing 40% of the rice that we consume, then we passed to produce only 10%. This just shows an example that agriculture is very vulnerable. There are different challenges that in this specific case of rice, it is like a good example that they can affect the production. Then we have the case of El Niño phenomenon. Actually, before this meeting, I was listening the news, taking the breakfast, and they were speaking about this because now it is a very important topic. And this news, although it is in Spanish, I just wanted to show you that is very recent news from this year, from this month. And mainly what it is saying is that the temperatures of the sea, they are increasing, and that they will increase above the average. And this means that then it will get warmer. And even they say that there will be other countries, including also India, that there will be this effect. So maybe also you are aware, and I was speaking with Aparna before the talk about the weather conditions. Currently in Costa Rica, the rain stopped a bit. In Costa Rica, we have mainly like 2 seasons, which means rainy season or dry season. So we are like entering in a phase of being like in a dry season, especially for the Pacific side and for the central part of the country. the Caribbean, it tends to rain much more. But this is a problem, especially for agriculture. So when we come back to the topic, we say this is something we really have to think and then to see how we can improve the systems. And today I want to show some of the projects that we have conducted and then some ideas that we can elaborate further when we open also the discussion. Also, I want to set a bit and to tell you where am I located. So I work for the University of Costa Rica. The University of Costa Rica, abbreviated UCR, it is a public university. We have 5 universities that are public in Costa Rica. UCR is the biggest. We have a population of students for the previous year for 44,000 students. And here in the map, again, you can see that the university is located across all the country, because the idea of the university is to provide opportunities to take academic degrees, not only for the central part of the country, but also in all the different regions. And in this way, also to support the development of the country. You can see that in San Jose, that it is the biggest campus. We have 32,000 students. So we are an university that is very active, and we work in different areas of knowledge. We work in arts and linguistics, basic sciences, social sciences, engineering, health and medicine, and agricultural sciences. That is the area where I work. In the case of agriculture, we are organized by the Faculty of Agriculture. Then the faculty is divided in 4 schools: animal sciences, plant sciences, where I belong, agricultural economics, and then food technology. From this, we have different research units. All of these bubbles that you can see here are different research units, which means experimental stations. We have crop protection research center, agronomics research center, molecular biology, in my case, seed and grain research, food technology, animal sciences, agricultural economics. We have an institute. We have one to deal with contamination. Those are the units. And we have also another players that they collaborate with also with the faculty, and that in this way it makes bigger all this network of collaboration inside just for the faculty of agriculture. And as I told you, we are a part of all this agricultural ecosystem that is located in the Faculty of Agriculture of Houston. In the case of seagrass, we work with grain technology, mainly grain quality testing. We work with seed testing. We actually are the official lab of Costa Rica for seed quality evaluation. We work with mycotoxins. We work with biotechnology. And with biotechnology, we have also established a good unit dealing with metabolite concentration quantification. And we work also with plant breeding. Our team is composed for 25 people. We work on teaching and graduate and postgraduate level. We work on research that I will show you some of the results that I think they are related with the topic that we're discussing today. And we conduct also outreach activities. We participate in the country in the processes of seed and grain quality control. For instance, there is a law that supports our role as official seed testing lab. This is relevant to mention because we provide services for public and private sector or to any person that just want to bring a sample to our lab. This also helps us to have income that we use for research. We collaborate also with grains, especially with the Ministry of Commerce. And then in the case of mycotoxins, we have norms for products that they are imported, that they cannot cross some levels of mycotoxins. Otherwise, they are not allowed to be to ingress the country to be commercialized. So we collaborate with the Ministry of Health and also with the phytosanitary system. And we have our assays accredited on ISO 1725. We do like a lot to do activities of outreach with farmers. Also, we have developed some specific products for schools and high schools that we call Germini kits. That is like germination kits, that are the kits for students to work with germination tests, but then to develop different experiments. But these pictures that you can see here are examples of trainings that we have conducted recently with different communities, and mainly trying to bring them information about seed and grain quality. So seagrass works with two main groups. One group dealing with technology of seeds and all the different aspects that it can involve, and the same it goes for the grain. And in this case, it is just one example of rice, but it is one of the crops that we work the most. In one of the experiments that we were doing, we wanted to get more information from the germination test, and then we also wanted to try to automatize some processes. And in this case, we started using digital images, mainly to quantify germination. And digital images, we did it with just a 2D camera and RGB camera, nothing that complicated. And mainly because with the segmentation process, that it is all this extraction of the different colors, it was possible to set when one seed was germinated based on the presence or not of the radicle. So this is actually physiological germination, just to see if there is emergence of the radical protrusion, and then we quantified in a manual way, an automatic way, then the relation between both, it was very promising. And from this, we conducted different experiments. In these experiments, the main advantages is that since we get images across the time, then we can end up having these germination curves. And in the germination curves, you can identify some samples like this one, but they are here located, that some they do germinate very fast and with high vigor, something like this here. And other ones that they germinate the same at 96 hours, but they do it slowly. So these are maybe crucial traits to quantify, especially when the plants, if they have to establish fast on the field, then they can get more values related with the germination speed. Also, in this figure that you can see here is that we were related the traits that we were quantifying manually. Here, these ones that I am just pointing here. And here on the right, the ones we quantified automatically. And then you can see that there is a relation with the maximum germination that is here with the count of normal seedlings. It is not that strong, but it's because the normal seedlings also quantification, it is different from the radical protrusion trait. And you can see here also we can evaluate, for instance, this T50, that it is a test that can be effectively used to measure also vigor of the seeds, and that it is the time to reach 50% of germination of the samples. With this, we can remember again the seed quality attributes, that in terms of seed quality, we are dealing with physiological, physical, genetic, and sanitary quality. And speaking about physiological, then we go to germination, dormancy, and vigor. In this case, I would like to point out to the importance of seed certification programs. And here on the right, what you can see, it is just a set of 2,000 random samples from data that we get in seagrass. And you can see that all these that are above the 80% germination percentage are samples that they did cross our standard for seed certification, that our standard is 80% for certified seed. And these are other samples that they didn't cross. And in some cases, some samples, they did show very low germination percentages. So in this case, seed certification program, I think it is something very relevant to promote, especially to try to promote, to have the highest quality seed, and this will translate into higher yields for different, what would be advantages that this seed will provide. One discussion that we have very common, it is something of this picture that you have, that you can see now, that there are two seedlings, and then the people will say, but the seedlings, they did germinate. But we try to make an emphasis that the case of the seedling of the left, it is what we call like a normal seedling, where you can see proper development of the root system, and it is in balance with the aerial part of the seedling. But in the case of the right, you can see that there are some problems with the root system. There are some colors that they might point to problems, maybe with pathogens or other circumstances. So that in this case, for us, it is a non-normal seedling. So in this case, it is something we do discuss quite often, especially when people, they have claims in our lab. So for this, the seed testing assay, this figure that you can see here on the right, it is in Spanish, but mainly my main argument to include it is that this is the beginning of receiving a sample, and this is the last step, just giving the report of how was the seed germination of the sample. And the thing is that something that is very simple, and one can say it is just to put a bunch of seeds to germinate and count them, it becomes something a bit complicated, especially to follow standards and then to make the test reproducible. So in this case, as I told you, we are accredited on ISO 17025. This means also we do metrological control of the equipment. We have to check for personal competence management, estimation of uncertainty, which means how much it can fail the results that we are providing, quality assurance of results and validation of methods, continuous improvement, risk management, customer service, audits, assessment by the accrediting body, and management reviews. We do participate very often also on rounds that they go international, where they release a sample, and then we participate to check how much we do deviate from what other labs they are also testing. When we go into these quality attributes, the other one is the physical. And in the case of the physical attributes, we speak about purity, humidity, mechanical damages, weight appearance. And this is one example of the purity analysis. And in this case, this is different parameters that we quantify. For instance, the first, it is just how pure the seed is. That in our case, that it is this standard, that it is a Central American standard. It indicates that the seed, for certified seeds, it should be above 98% of purity. Then there can be inner matter, there can be other seeds, there can be red rice. The red rice, it is a problem here in our country. There are weeds that in some cases, there are weeds that should not be tolerated, otherwise that they are wild plants that they are growing. Then there can be presence of other crops, insects, and then humidity content. At some point, we did also some tests, and this is data from Taylor Scott from Skyway Analytics, and we were collaborating with him because he was taking some pictures of sample of us using this type of equipment. But this type of equipment, it takes multispectral images. That it is a very interesting technology because in this case, with RGB images, we see just in the three colors that the camera provides. But in this case, then it can go to different wavelengths that can provide or give us extra information. But also, this helps to make automatization, especially to count for full seeds, and then to count for damages, to count for other, what would be matter that is present on the samples, and even other seeds that are related. So it is an interesting technology that one also can use as a type to innovate on seed quality testing. With the communities, when we do trainings, one of the kits that we bring is, and the question that we ask them is, how is the humidity on your seed lot? And then the people tell us like, but Liz, my humidity is very good. My seeds are dry. And then we ask like, okay, maybe we didn't ask the proper question. That maybe the proper question is like, how is the most moisture content in your seed lot? Because it has happened for some communities of us that the seed looks perfect, but in reality, then the seed has problems or it has very low germination, especially with seed communities. When I speak about seed communities, is that we have different people that they grow their own seeds, and from their own seeds they produce their own food. They work with, for instance, maize, beans. They produce their own rice. They have also cassava. They have taro. They have different crops that they have in their farms that they are not that small, but then they do grow, and in that way they have a balanced system. So in this case, we provide them a specific tool because they were not quantifying the humidity content. And we provide this, that it is a ring and it has a moisture indicator that when it has high humidity, that it is in most of the cases where in the farms that we visited, it has this pink color, and when it is dry, then it is a blue color. And something else that we provided them and that we learned from the University of California, Davis, it is the use of cell light. All this technology is related with a concept that is called dry chain. You might be aware of it, but also there is this publication where we participated showing cases of different parts of the world. This technology we learned through Ken Bradford, and also Johan van Asbroek help us a lot, and it is something very interesting to work on. These are examples of cases of Costa Rica where you can see a huge diversity, and I would say diversity, it is something very relevant that we have also to take care in case of speaking of climate preparation, and then also having a diversity for issues that come in the future, and then to have resources for plant breeding. And these are examples of diversity that the communities, they do have. Here in Costa Rica, we have people that they love having diversity in their houses. These are cases of even like a type of sanctuary of seeds that a community has, where they have all the different genotypes that they do store. So in this case, for us, for this type of people, it is relevant to learn how to process the seeds and decrease humidity content. So we provide also training, and this is just an example of something that we provide them. That the first step is to know how is the moisture content of the seeds. The next step, it is that if it is pink, then add zeolite and have it for 2 days with the zeolite. And after that, check for the humidity again. And if the humidity is right, then store your seeds. So this has helped a lot for the humidities, especially to improve their germination rates and then to not lose diversity. So when it comes to seed lots, for me, it is very interesting because a seed lot can look perfectly, but you really don't know if it will be able to germinate or if it will have another problems on this. And this also can get complicated, especially with the sanitary part, because the idea is that it is healthy and pathogen-free. And for that, then you must check if there are diseases on the seeds. But something that we are working on, or especially a professor from us, and his name is Andres Hernandez, and he is from the Seeds and Biotechnology Labs. And it is something that I consider very relevant and that is very innovative, and is delivering microorganisms via seeds. And in this case, his project that is called micromaize, that is like micromaize or with micromaize, is that he has a hypothesis that is to isolate microorganisms. That is something that we are using a lot on the country. And then you inoculate these consortia or groups of microorganisms on the mother plant. And then once it is inoculated, the idea is that there will be transmission of the microorganisms to the mother plant. and then that this will be checked on, in this case, rhizotrons, because he is interested not only to see on the upper part of the plant, but also on the benefits for the root system. This is something that we are currently working on, and to hopefully provide benefits when the seeds are prepared. When it goes to genetics, variety, purity, and then also the attributes that the genetics can have, we all do know, and I think it is one of the strong points of ATPBR, is that through the variety, we can release different genes that they are crucial for different aspects, and then also for dealing with biotic or abiotic problems. This also, in the way of genetics, it help us, I would consider, to bring also attributes related with seed value. Because on the left, we already discussed quality attributes, but I like also to speak about seed value. And in the case, for instance, some of the things that give value to the seed, it is the yield that it can provide, but there are also nutritional aspects, there are also commercial aspects, and cultural aspects, that all of them are relevant. These are examples of rice that I am not giving like advertise or something on any brand from Costa Rica. These are just a mix of pictures that I took recently from the supermarket. And these are examples of rice that you can see here that this is a full brand, or this one, it is a milled rice. Here it says that it is free of pesticides. Also, our rice in the country, it is fortified, which means that nutrients are added to the rice in order to make it more nutritional for the people. Here there is a logo of neutral carbon, and then here are minerals and vitamins also that are added. So are examples of this, or this other one, you can see that it is organic. Here there is another logo that it shows for pesticide free. So this shows also varieties that they were adapted for this specific commercial system. And here you can see also other trait of our rice is that here this 99 it means that the rice inside the package is 99 full rice. And that is here this other one, for instance, it says 95. This is other of the traits, other traits that is used here in Costa Rica to set up a price for the rice that is consumed. Something that we have been working on, it is on quantification of vitamins. And for instance, vitamin E, it is a relevant antioxidant that is not only good for the health of the seed, but also for us when we consume it. This is an example, and this we quantify using HPLC, the liquid chromatography. And here you can see a sample of rice, and here you can see the standards. And each of these peaks, it shows different tocotrienols or tocopherols that are forms of vitamin E, and then that we can quantify. On this table on the right, you can see that there are different types of rice that we have on our germplasm, and that you can see the quantifications of alpha tocopherol and gamma tocotrienol and total vitamin E. And the relevant thing is that you can see that some of these genotypes, this is just a summary of different that we quantified, they have high levels of vitamin E. Then that means that when we want to add, it is not only more food, but also more nutritional food. And this is a way also to check for this. We also follow this process further, and then we recently also published this paper where we were looking for a specific gene that we consider can be involved with the quantification of or dealing with these vitamin E concentrations. We follow a previous paper that they were pointing to this specific region, and from this we developed HRM markers that in the markers, in this case, we were detecting this polymorphism that you can see here of having a double G or a double A. And in the markers, then with HRM, we can quantify very properly these differences. But when we were studying this, we also identified that there was a big gap on the gene. Here you can see the model of the gene, and then the gap that was present. And in this case, also we confirmed it with regular PCR, and then we observed these differences. After that, we also quantified, again, these vitamin E forms, and in some cases, there were relevant differences, but then we are doing more research to see if these differences are mainly attributing to these specific polymorphisms, or if there are other regions that they are involved. For us, this is important because molecular markers, it is very relevant for advance in plant breeding, but they also can provide data for nutritional aspects of the varieties that they are grown in the country. We have one professor, her name is Andrea Diaz, and she's from the biotechnology lab, and she recently published this paper, and it is something that we are also moving on, working more on this part of dealing with the nutritional part of the grains that are consumed, or the varieties that are released. And in this case, here this is the map of Costa Rica, and this is a region on the Caribbean side where rice was produced, and then we have indica, aromatic, and japonica rice, and they were studying how was especially the absorption of the rice bran compared with a milled rice. And they were using a system that is an in vitro digestion, and mainly they mimic what happens on the mouth, the stomach, and the intestine. And they use a cell culture that is called CACO2. And in this case, it mimics how then the nutrients are absorbed. And in this figure, what you can see is that there are two compounds. One is called ferulic acid, and another one is gamma oryzanol. And what you can see here is that there are different genotypes. And then something relevant is that ferulic acid had a significant uptake. For instance, you can see that it goes up to 20%. In the case of this gamma oryzanol, it was a moderate absorption. But also something very relevant that I was discussing with this researcher, it is that it's very, very interesting to see that there are some specific genotypes where the absorption of the compounds, then it is higher. So it means that it should be possible also to select for genotypes that they are producing specific genes that they will also help making more uptake of the nutrients. And in this case, to make it better for the population. Despite all this that we have discussed, getting on the last part of my talk, it is that despite all of this, we haven't adopted certified seed in all the regions. And when using certified seed, we should be speaking about increasing yield, because then there will be higher germination percentage and also higher vigor. There will be a better crop synchronization. We should reduce use of agricultural inputs, herbicides, fungicides, or reseeding. And all of this, then it is mainly related because using low quality seed can produce risk. We have one example, and it is the Bean Plant Breeding Program, and it is a program conducted with one professor from UCR, Nestor Chavez, and one of the person that works for a governmental institution that is called INTA here in Costa Rica, and they work with crop innovation, Juan Carlos Hernandez, and then they run the legume program. They work on the scheme of participatory plant breeding, and for me this is very relevant because since the farmers, they provide feedback on what they are looking on the new varieties or advanced lines, then they can provide really true information of what will be the main attributes that can be selected and then releasing new varieties. This program, once a line is selected, they provide the foundation seed, and in this way they do select for the different varieties and then provide it for the farmers. We have other group of researchers, and in this case, it is a group led by a colleague of mine that is called Victor Rodriguez, and this is also his email over there. And they are working on data that it is not published yet, but mainly what they tried, it was working with farms that they use safe seed compared with certified seed, and they quantified the yield. This is the profit, and this is the difference. And they work on 3 field inputs. Field inputs will mean when it is low inputs, which means you add very few products, then have medium and high. And high means that you add all fertilization that you can provide, all applications that you can do, responding to different necessities of the crop. And in this case, to make the long story short, you can see that it can range on differences that they can go from 3.2 to 14.3 increases of yield. This translated on our currency and showing it in US dollars, it can go from $190 to up to more than $1,000. And then this will show differences from 19 to 64%. So this shows data that has been provided in the case of the certified bean program to try to promote the use of certified seed. And to be closing, something that we are working on, it is on a network of soybean, and we are not like soybean producers. We mainly import all of our soybean, but through the different problems that they have come in the recent years, pandemics, container crisis, weather differences, then different people have asked us to have seed varieties for soybean. And this, we work together with groups from universities of United States and also from NGO from Canada that works in Guatemala. And we started then doing trials with different lines of soybean. These are soybeans that are grown on the Caribbean side of the country. And through this necessity, then we started working with this network. You can see here one example, and this is the farm. The picture, it is a farm that it looks that steep. I mean, there are farmers that they grow on mountains, the products. Here you can see some soybeans. Here there are beans, and in some part there were maize. And these are true examples of production systems that we have in Costa Rica. And in this case, the soybeans, it is our plant that they can fix nitrogen. Then you can rotate with other crops. In this way also, they are very resilient in our case, especially for dealing with water shorts, especially when the plants are growing. So then we are promoting the use of these soybean. And this is a recent trial and a field day that we conducted, and it was at the city. And then we with farmers and also people interested in soybean. The idea is that to provide food security and also for exchange of communication and also on academic and scientific collaboration. We have different focus areas, for instance, seed production, to deal with soybean training, competitiveness, which means products, tofu, intersectoral alliances, and also public advocacy. Just to start finishing my talk, something else that we have participated is that Costa Rica has a national seed policy, and this policy actually started from something from Tigras, that it was one of the main actors, but also it was working with farms, public sector, with the National Seed Office, and then different actors that at the end provided a national seed policy. We have different technologies, also working with biotechnology, also working with all the topic of mycotoxins is very relevant, and it is also very broad. We have some work of plant breeding, in our case of seagrass, working mainly with soybean and rice. And also we have a unit that did work with crops that are underutilized, because they are crops that they are like native crops that are very relevant for nutrition, but that people don't use that much. So we are doing research to see how we can provide new varieties. And something also we are starting to explore, and I think it is something that for plant breeding has to be considered, is the use of systems. This is a agrivoltaic system we are starting. This is a recent picture. And mainly it is a system where energy will be provided on the top, and then the plants will grow down. But this means also that varieties, they have adapted for this. So it will make also a challenge, especially for crops that are related, like more horticultural crops, vegetables, this type of crops that then can be tested under these schemes. And finally, I would say that climate resilient seed systems and innovations really deal with collaboration and also exchange of knowledge. So that's why I really appreciate the invitation of ATPBR to share with you this night, and in the case of mine this morning, in order to discuss a bit of different topics that can help us. Thanks again, and I showed all these different data from different people that I collaborate with, and then thank you very much for your attention, and I am open then to start the questions session.
Thank you very much, Luis. I have been in your country, Costa Rica, and I have a taste of gallo pinto, casado, arroz con vegetales, yuca frita, and sopa negra. Did I get them right?
Yes.
Yeah, it was a beautiful talk. I have also tried to say something. I've tried to say something to you in your language. Let me
Estimado Luis, muchas gracias por su charla tan iluminadora y enriquecedora en el webinar de ATPBR. Su conocimiento, claridad y valiosas perspectivas hicieron que la sesión fuera sumamente informativa e inspiradora. Apreciamos sinceramente el tiempo y el esfuerzo que dedicó a compartir su experiencia con nosotros. Con gratitud y mejores deseos.
I think I'll read the English version for the benefit of others. I just said, dear Luis, thank you very much for your illuminating and enlightening talk at the ATPBR webinar. Your knowledge, clarity, and valuable insights made the session highly informative and inspiring. We sincerely appreciate the time and effort you devoted to sharing your expertise with us. Thank you very much. With that, I would like to thank you again for your time and efforts. Very good presentation, compliments. And now I think we can open the session for questions. If people have questions, please do ask. Yeah. I expect a lot of questions because we are about 30 people here, and I'm sure, as I said, this is a weekend, but still you have so many people. That's interesting. Yeah. Aparna, you wanted to say something? Can't hear you. Your voice is breaking. Yeah. Please. Okay. Yeah, it is glad to have you with us, and we have something in common. Aparna was also in Netherlands. She studied at Wageningen. I think the question, if possible, please give PP. Mohammed PP. What is PowerPoint? Yeah, Dr. Shankaran has a question. Dr. Shankaran, please. Please unmute yourself.
Am I audible?
Yes, sir. You are audible. Please go ahead.
Is certification, seed certification in Costa Rica is voluntary or compulsory under the legal provisions? That is, anybody producing seed has to have it certified, then only it can be used, or uncertified, what we call in India as a label seed. It's a voluntary. That means certification, official certification is not compulsory in India. What's the situation in Costa Rica?
Yeah, before you answer,
I-
Louis, 1 minute. Dr. Vyambu Shankaran, he was my senior in post-graduation at Coimbatore. He's regarded as the very prominent seed person of India. He worked with National Seeds Corporation and is seed technologist by training and PhD in seed technology. So that's his question. Please go ahead and answer. Yeah.
Voluntary. There are countries.
Your voice is getting voice is breaking, Luis.
Yes, I can.
Your voice is breaking. Some connectivity problem, apparently?
Can you hear me there?
Yeah, now it's clear. Now it is clear. Go ahead, please.
Okay. That it is voluntary. We have discussed this, and there are some countries where it is obligatory, mandatory, and it is voluntary. There were some very good years that it was above 90% of usage of certified seed for some crops like rice, but other years that then it decreased because when there are shortages, what the farmer will do is to not use certified seed. That is something that for us it will not make that sense, but they are the people that they are producing. So that is the first thing that they do, and yeah, it is like that. We have discussed it, but we don't have like any movement right now to make it mandatory. Not in India. How is the situation? It's mandatory or?
Earlier, as I said, the organization, huge organization with which Dr. Shankaran was associated, National Seeds Corporation, they have the certification process. Earlier, we were getting certified seed. There are state seed corporations, NSC, maybe he will add to what I am saying now. And private sector, we sell what we call as truthfully labeled seeds. It is TFL. That means we guarantee the quality and purity of the seed. We call them truthfully labeled. Dr. Shankaran, you would like to add something? I think Pragadishwar wants to say something.
That's fine.
Yeah. Dr. Shankaran?
Yeah.
Want to add something, sir?
Pardon?
You want to add something to what I said?
No, I wanted to know. See, in India, we have a lot of talk about the traditional varieties, indigenous varieties of various crops, especially starting with the wheat or paddy, etc., important crops. And India has a very wide biodiversity, and therefore, and it's very agroclimatic zones are plenty, and therefore you have a wide variety of germplasm of traditional and indigenous varieties. What is the situation? And now a lot of talk is going on. Apart from the planned breeding programs, the traditional varieties are also.
Pragadishwar, please mute yourself.
Importance now. Is the situation same in Costa Rica? Or you have a traditional varieties or all varieties developed by the planned crop breeding program? How do we compare it with India?
Yeah, that was the question. Luis, you can answer it.
Yes, well, there was a bit of background when he was asking the question, but from what I understood is that in India there are different types of seeds, traditional seeds, and we have something similar here in Costa Rica. I would say that it is something that is called like heirloom varieties. For instance, that there are varieties that the families have gotten or communities, and they pass it from generation to generation to generation.
Yeah.
That is like one group of seeds that we have. We have also the varieties that they are registered and that they are registered on the National Seed Office, and in that ones they get on the formal system, and they can be commercialized. And we have the other one that will be the certified system. That in the certified system, there will be also genetic seed, and there will be foundation seed, and then there will be different processes. But then, just to answer, we do have that, and we have varieties that also they are like native, and we have also biodiversity law, and they also somehow protect the use of these varieties. But when it is for the own consumption, it will be not a problem. When if they will be used for commercial purposes, then there is a specific office where the people then can approach, and then they can get permits for using these varieties.
Does that answer your question, Shankaran sir?
The last one, sorry. What is the common seed vigor test that we try? I mean, we have succeeded in paddy, or what are the very prominent vigor tests which you are feeling quite good to judge the seed vigor of different crops? What are the common tests that we have? Because ISTA has prescribed several or recommended several vigor tests for different crops, and we try different methods, and finally I feel the speed of germination is one. Somebody will say the dry seed weight, dry seedling weight, different components are there, accelerated test, etc., etc. So what is the vigor test which you have found to be quite revealing? Say, as an example, in paddy, because you have talked about paddy and soybean a lot being. So what are the vigor tests that you have found it to be quite good, revealing?
Sharon, can you reformulate the question? It's because I couldn't.
No, he was
properly?
asking, that Shankaran was asking about seed vigor test. He was saying germination, how do we measure whether a particular variety seed is vigorous or not? Seed vigor. Did you get it?
No, I still don't.
Vigor is how healthy the seed is, how fast it can grow, and that's called vigor seed basically. Vigor is when it grows healthy, it grows fast, it grows quickly and healthy. That is called seed vigor. So here he was telling about the seed vigor tests in India, where we just take a germination and how fast the seed grows and how fast the roots and stem elongate. Similarly, what vigor tests do you have in Costa Rica? That was his question.
Okay.
Right, Shankaran
Yeah,
sir?
thank you.
Very correct, very
Yeah.
correct. Yeah.
Yeah.
Because in one of the
Yeah,
slides,
thank you.
you have mentioned about physical purity,
Yeah.
genetic purity, varietal purity, and seed vigor, physiological
Mhm.
aspect of quality. So
Yes.
we quite
Yes.
often find it difficult to pick up one or two for different crops. You know, so I was just thinking your experience may have some value for us.
Yeah.
Thank you.
Got the question,
Well,
Luis?
yes, we receive samples from all the different crops that they are commercialized in the country. We produce few seeds that are local, but for instance, when it comes for vegetables, most of our seeds are imported. And then we work with more than 100 species. But for this, the main test, and then they are included in the law, it is the purity analysis and the physiological seed germination analysis. So those are the two tests that are mandatory for the people that is on all these commercial formal process. We do provide other tests. In Seagrass, we have more than 38 tests that we provide, and some they deal with seed aging. Other ones, it is working with a bigger tests, speed of germination. Then we have also humidity content quantifications. That is also something that is included. We have the quantification of plant hormones on using HPLC. Those are the highest tech traits that we have available. But I would say mainly most people will go just for the seed purity and seed germination.
Okay. Okay.
Thank you. Thank you.
Thanks, Luis.
Welcome.
I think Pragadishwar was trying to make a point. He wrote something. You can ask yourself. You can unmute and ask Dr. Pragadishwar. Are you there? Please unmute
No, can you
yourself.
repeat?
Yeah, he was writing. Can you read that, Aparna, if that is there in your chat box? He asked a question. He recently It's a bacterial disease.
Yes, thank you. In that case, I would say I cannot provide like any recommendation. I'm sorry. In our case, we do check for the seed to not have problems. If it has problems, we have an unit that it deals with crop protection. So then we present the samples for them to identify what is going on. But we don't have, or at least I cannot provide any suggestion in this case. I'm sorry. Something else that I can check here on the comments is that yes, I can provide the presentation. I can send it to Aparna, and then you are free to share it with the people. Yeah.
Thank you. Thank you.
Yeah.
Hey, what you're saying is right, Luis, because bacterial diseases, we find it difficult to manage and control. And what we have found in tomato and peppers is bacterial wilt resistant varieties and hybrids. That's one solution we are using for control or management of bacterial diseases that have to control.
Yeah, here it happens once also a big problem with melon seed some time ago, and also it was dealing with bacteria. But yeah, I will try to look for more information and also to learn for myself. But yeah, it can get complicated.
Was it bacterial blight?
I don't remember. It was many time ago. I was a student at that time.
Okay. Yeah. I think any other questions? Anybody else? Aparna, it's time to close. Yeah. Thank you very much, Luis. Once again, it was very enlightening, and thanks for bringing all this knowledge from Costa Rica. I was in Costa Rica in 2015. And we spent about 4 or 5 days going around different places, looking at your beautiful forests, well-kept environs. Great. It was nice having you with us. So please do keep in touch with us. Be associated with what we are doing. Try to help us to spread the knowledge. As Aparna was mentioning, we do more of capacity building and training. As she was mentioning, we also have written a book, Market-Driven Plant Breeding. You can find it on the web. If you want, we can also share the details of that book, where we have tried to especially mention the specific aspects about private sector in India, seed sector, and also global perspective. Yeah.
Thank you so much.
Yeah. Shankar is saying thank you so much. Thank you, sir. Thanks for joining. And
thank you so much. It's a pleasure. It's a good opportunity to hear about Costa Rica.
Yeah.
Thank you so much.
Okay,
thank you very much for you.
Is it weekend for you? Is it holiday for you today?
Yes. Now it is a weekend, and also for you, have a good rest. Also, a nice weekend.
Yeah. Now it is. I think it's dinner time for us. 8 o'clock. We have our dinner around 8:30. So muchas gracias once again. So thank you very much. Okay. Bye.
Thanks to you. See you.
Bye.
Thank you, partner.
Yeah. Bye.
Bye. Thank you. Thank you.